Um, Hi, I'm Ken. First of all, I would like to thank the organizers for inviting me. It's a really great pleasure to be here. I remember my first time attending this free energy workshop back in maybe 2015 or so, and I was sitting somewhere in the audience and really overwhelmed by all the presentations, and never thought that I will be standing here myself giving a talk today. So today I would like to share some of the work that I have been doing when I was visiting Memorial Sloan Kettering Cancer Center when I was inside Jean Correa's lab. As you can see, my topic is about force field development using ML-based methods using large-scale quantum chemical dataset. So I would like to get started through a very simple slide and some thoughts about the challenges and perhaps the considerations for running free energy calculations. As we all know, sampling issues is one of the critical aspects in getting your free energy calculations to run. Also, initial post generation is also a very important aspect. It's not just about how you generate your ligand poses, but it's about how you define your protonation states for your entire system. We also had a talk about the transformation networks for the ligands yesterday. Designing your transformation network is also not trivial. And also, we need to have a reliable physical model to run your simulation reliably and leading to accurate free energy calculations. My main topic, as I said, is about the force field development here. One of the widely used physical models, perhaps today, is a MM force field, which characterizes the energy landscape of molecular systems through a very simple functional form. They are really oversimplified and inaccurate, but seems to work in practice, and that's why I think everybody likes to use it. But when we look into how these force fields are developed, it's really difficult. It requires a lot of human effort where we have to kind of split our force fields. We have to split our development based on different biomolecular species. And we intend these to be compatible, but it's not co-optimized, which may require a significant effort to extend to new chemical domains, which could be really challenging if you're running a drug discovery campaign. For example, you could have like a peptide system as your drug target, or as a drug, but you could also have a chemical modification to your peptides, which makes your simulation run very difficult. So my motivation here was, how can we develop a self-consistent MM force field? Meaning that how can we create a MM force field that is applicable to multiple biomolecular species using a single MM force field that is robust and easy to extend and perhaps more accurate to run a reliable free energy calculation. So in the lab, we have been working on a machine learning framework to do that. We call it Esploma. But before going inside what Esploma is, I just want to go back one step and rethink about how this MM force field is developed in practice. There are multiple ways of developing this force field, but I guess one of the widely used approaches is the atom typing scheme. What this is that we define an atom type for each atom. You could have, for example, a carbon atom, which could have a sp2 characteristics, which could be bonded to a oxygen and sulfur atom through a double bond. But you could also have a same carbon atom that has a sp3 and attached to a hydrogen atom, which means that even though you have the same carbon atom, the atom type will be different, assigned with a different force field parameter. When we revisit how this atom scheme works, we can kind of rethink of this as a graph neural network architecture. So that is one of the keys in the Esploma architecture. So this was there's a very nice package called Esploma, which was developed by a former grad student in the lab, which was led by Yanjing Wang, where we used a graph neural network to come up with atom embeddings, and then we can pull them together in a symmetry-preserving way to come up with a torsion at angle and bond embeddings, which can be fed further forward into a neural network to come up with torsion, angle, and bond parameters. With that, we can compute the energies and the forces as well. And we can use a reference data, for example, a quantum chemical data with the energies and forces, and try to minimize the discrepancies and update and optimize the ML model itself. The training itself could be done only once, and once we have a trained model, we can apply this Esploma architecture to parameterize any kind of a biomolecular system. So the proof of concept and the initial results were published in 2022. And when I first joined the lab, I thought this was really kind of exciting. But I had one question, that how applicable is this to real drug discovery campaigns? So what I had been doing in the lab was that I was revisiting this Esploma framework and trying to retrain this Esploma using a larger quantum chemical data set that we can grab at that time. So this is kind of a overview of what I have been doing over the past 1 or 2 years. In this study, what we did was we curated a quantum chemical data set that consists about 17,000 unique molecules with over 1.1 million conformations, which consisted small molecules, peptides, and nucleic acids. Each conformations were derived using a different kind of method. One could be like MD snapshots that store relatively higher energies. There are other conformations that has been generated using a DFT optimization trajectories and also some torsion scans as well. So it consisted of really a diverse data set. We also kind of revisited how the Esploma framework by, for example, looking into the hyperparameters, introducing some force matching into the protocol, and revisiting the regularization scheme to generalize the model better. We also kind of invested a lot of time in benchmarking the developed force field. Ultimately, we want to use this for free energy calculations, but I wanted to make sure that I get the right results for the right reason. So we kind of conducted several different kind of benchmark studies, just focusing on the small molecules and also like peptides and proteins as well. I should note that in this study, we only focused on the valence terms. So we started off from a small molecule class 1 force field, the OpenFF 2.0, and then try to extend that to other biomolecular species. So this is a very, very, very busy slide, but one of the simplest, I guess, the initial benchmark studies that people will do is just look at the RMC metrics of the energies and the forces that is trained. The takeaway message here is that we were able to reproduce the quantum chemical energetic properties more accurately than the baseline force fields. But honestly, this isn't really, convincing, not very informative in terms of how reliable the force field itself is. But it's like a control experiment that we do get low numbers suggesting that the training itself were formed well. So what we did next was we kind of dig into actual molecules and see how they performed. We first looked into the small molecule geometry benchmark data set, which was led by Pavan Behara, a former member of the Open Force Field member. Here what we did was we grabbed a out of distribution data set, the OpenFF industry benchmark data set, which consisted about 9,700 unique molecules with 73,000 conformations. What this is it's a set of small molecules shared by the industry partners that are relevant to their interest. And what this data set holds is that there's a QM optimized geometries for these small molecules. And what we wanted to see was that when we start off from a QM optimized geometries and minimize using one of the MM force fields, how well can we preserve its geometries? So that's shown on the right-hand side. The machine learned force field, we call it Esploma 0.3, and compared it with some of these small molecule force fields. The overall trend, we do seem to be compatible or sometimes better for all 4 valence terms, but we do see some outliers for the bonds and the angles. So we need to take a deeper look in what's going on here. So all these outliers were coming out from a silicon amide. Particularly speaking, we saw a elongated S-N bond distances, and also a non-planarity in the pyrazole and the imidazole silicon amides. So this is one of the limitations of our current Esploma force field, and something that should be kept in mind, that we might see unexpected behavior if you're handling with a silicon amide. So what we did next was we moved on to a different system, slightly a little bit more complicated. We tried to reproduce the condensed phase properties of short peptides, which was led by Chapin Cavender, another member of the open force field. And as a comparison, and I should say that we were trying to estimate the NMR experimental scalar couplings using one of those force fields. As a baseline, we compared it with the Amber 14SB, and we found that we were able to reproduce the NMR scalar couplings better than the well-established Amber force field, which was really kind of exciting to see. So I was really happy. We were really happy, and then moved on to a slightly more difficult benchmark data set. We tried to now reproduce the condensed phase properties of the folded proteins, which was led by Annika Friedman from the Michael Schultz group. Here, I should kind of explain what ANE is. It's an average normalized error, which is somewhat similar to the chi-square, but it's a normalized value ranging from 0 to 1, where 0 means a better fit to the experimental value. The overall performance for Esploma 0.3, we were able to somewhat reproduce the NMR experiments, but less accurately than Amber 14SB. But still, this was kind of encouraging to see that we are able to reproduce the condensed phase properties of the folded proteins. And we ran this simulation for 10 microseconds without blowing up the system, which is also encouraging. So with all the benchmark studies that we have conducted, starting from the small molecules and the peptides and proteins, we next question ourselves, can we run a simple long simulation where we parameterize the protein and ligand self-consistently using this Esploma force field and see if we could run a stable simulation. So that's what we did here. We used a TYK2 protein ligand complex system as our test system. And we tested two different parameterization schemes. One is that we parameterized the protein ligand using Esploma 0.3. And as a comparison, we used Amber 14SB for the protein and OpenFF 2.1 for the ligand. And we monitored the RMSD throughout 3 microseconds of simulation. And we were able to see that we were able to simulate a stable simulation when self-consistently parameterizing the protein ligand with our Esploma 0.3. So with all these benchmark studies, we were kind of, okay, we have a machine learned MM force field that seems to work with all the benchmark studies that we did. So as our final experiment, we moved on to this protein ligand binding free energy benchmark study. In this study, we tested against 4 target systems, TIC2, CDK2, P38, and MCL1. All these data sets were extracted from the original open force field benchmark suite, but I did some slight modifications to the ligand poses and the transformation networks, because for the ligand poses, I wanted to make sure that the initial poses were reasonable from a expert point of view. And also, I wanted to make the transformation networks as simple as possible, just in case that I see a weird behavior in the free energy calculations, and I can easily go back and troubleshoot what's going on. And this is the result that I get. All the free energy calculations were ran by Percy's 0.10.1 with a slight customized version to allow the simulation with Esploma 0.3. The right-hand side is the result using Esploma 0.3, where the protein and ligand is self-consistently parameterized. As a comparison, we ran the same free energy benchmark study using the same initial poses and the ligand transformation using Amber force field and OpenFF 2.1. The middle panel is kind of a hybrid system where we use the Amber force field for the proteins and Amber force field for the proteins and Esploma 0.3 for the ligands. The performance using Esploma 0.3 was somewhat compatible with the baseline results. We weren't able to really see a major increase in the accuracy, but we were really happy to see that we were able to do as compatible as the well-established methods, suggesting that we could probably aim for a more difficult target like covalent inhibitors and perhaps like a peptide with non-natural amino acids. I should also note that the ligands from the protein ligand binding free energy data set is quite dissimilar from those that are used to train the Esploma. The maximum similarity between these two different data sources were about 0.5 using the Morgan fingerprint. So this kind of demonstrates the generalizability of the Esploma architecture. Yeah, so the Esploma package itself is open sourced. And also, I was trying to work on this so that, Esploma is for everyone, and wanted to make this training protocol really easy as much as possible. So you could train your own Esploma force field in a few lines of codes and run some simulations using Esploma, using this wrapper called SFIT. So in conclusion, I have developed a machine learned MM force field that leverages large-scale quantum chemical data set, which is really easy to extend to new chemical domains of interest. I have benchmarked this machine learned force field against different benchmark studies, and I personally think that it's applicable for free drug discovery applications, not just for free energy calculations, but maybe if you have a target system that is not easy to parameterize using your existing force fields, maybe this could be a alternative. Yeah, you could run it with other popular MD engines as well. And the Esploma architecture is really highly flexible. In this study, we used a class 1 functional form, but it could be extended to class 2 and other kind of functional forms as well. We also had a talk from yesterday about learning from free energy calculations, and I think this is another area that we could further extend the research, not just by training from a quantum chemical data set, but leveraging experimental results as well. We could also go for evaluate the model uncertainty of the force field and see how that propagates with the free energy errors, or if there are errors or some outliers. Yes, with that, I would like to acknowledge everyone who were involved in this ISPLMA 0.3 study. And I would like to especially thank the open force field members because I couldn't have done this research this quickly without the open access to all the quantum chemical data sets that they have generated in the past. And also we leveraged a lot of the infrastructures developed within the consortium to move this project forward. Yes, that will be it. And thank you for your attention.
Thank you again for a great talk. We have now time for a few questions. There's our first question, I think, in the phone. Yeah.
Thanks, Ken. It was a very interesting field, and you probably mentioned it a bit like, okay, one of the limitations might be the similarity of my training set to the ligands that I used it for. So do you have plans to assess the chemical similarity and grow the chemical diversity of your 17,000 molecules in the QN calculations? Do you think this will improve the results?
I hope it will improve the results. One of the open questions is what kind of data set is important to make this ISPLMA model more generalizable and more accurate. I think it's not just about the chemical topology, but it also matters about the geometry. So in this study, we used MD snapshots, some DFT optimized trajectories. But the question that I still have is that I'm not sure what is the right balance between having a high energy confirmation in your data set. Probably we also need a low energy confirmation in our data set. So I think it's a balance between the right geometry confirmations inside our data set and also the topology to further improve this model.
Okay, we move to our second question.
Hi, amazing talk. Can you do covalently bound ligand to the proteins? Have you benchmarked against it?
So covalent bond inhibitors.
Oh, yeah.
In principle, it should work, but I haven't benchmarked any data set. If you have any interesting benchmark data set that can be run for covalent inhibitors, I would like to try and see myself how it works. I have my fingers crossed that it will work, but much easier than the other force field packages.
Okay. So thank you again, Ken, for a great talk.
Thank you.